Welcome back to our GCP-Mindset channel and to part 2 of our web seminar of sample size calculation in clinical trials. In part 2, we talk about the role of the sample size calculation throughout all study phases.
In the second part, we will have a look at the sample size and its role throughout the study phases. And first of all, what Anja already mentioned is that in clinical trials, the different phases have very different objectives. So it's very important if you're in a phase 1 study or in a phase 4 study. And the first thing we should notice is that the trial objective and the sample size justification must always match. This is important. The second thing is that when the drug development proceed and we obtain more evidence over the time, then this evidence should always be included when we do a sample size calculation. For example, as you remember that when we have to make the sample size calculation, we have to assume the treatment effect and the variance. And when we have already published information about the treatment effect, when we're, for example, in a phase 4 study, then we have very good estimates for the variance and also for the treatment effect we can plug in our formulas. And yeah, the last point is that there are also several types of clinical studies. We have drug trials, we have medical devices, we have IVD trials, and all they have very different requirements. And there is also the possibility really to optimize the respective requirements there. For example, it's very important that we select the correct endpoint and how we collect the endpoint. But we will come to this later. We have a work example and we want to go with that work example through all the study phases. And we have that a researcher had an idea how the blood pressure could be reduced in subjects who suffers from hypertension. And this researcher has then invented the so-called theta blocker. And what we now see is which phases this drug has to go through until it is available in the stores. And also we will see what role does the sample size have in all these different phases. So let's start with a brief summary of the drug development phases. We start with the preclinical phase where we test our drug in animal models to see if we can find really an effect. If we are found an effect, then we will go into the phase 1 study where we first test our drug in the human body, and here we will focus on the safety profile. When we agree on that the drug is safe, then we are more interested in the efficacy and what kind of dose we should use and what is the optimal dose. So we make a proof of concept. Is there really an effect that we can observe, and how high is the effect? And also in which dose is the most preferable one. When we have enough information and feel confident to go to get the mark approval, then we will have this phase 3 study, which is called the proof of efficacy study, where if we conduct this study successfully, we will get the market approval. And if our drug is already on the market, then we will also have a post-market surveillance that we have a look at more rare diseases and also to explore a little bit more the treatment effect observed in real-world settings. When we are in a medical device setting, then we would have just the pre-market study. This would be then a phase 3 study comparable and a post-market study that is compared with the phase 4 study. So let's start with the phases. So we have this preclinical phase where the objective of this trial is that we want to see an effect at least in an animal model. And we want to prove that there's an effect, and therefore we have to use a confirmatory approach. But there is a problem in a preclinical trial because as this medication is never tested before in an animal. So we don't know the effect. We neither know the variance, and as you can see that we have several doses and several time points and sometimes also several type of mice, we also have a multiple test problem. For example, when we have just 6 doses, 3 time points, and 3 type of mice, we will have 45 groups we want to investigate. And when we then remember that we have to make some assumptions regarding the treatment effect, then we and also have to estimate the variance, we will have 55 assumptions we must do prior to the study, although the medication is never tested in any body before. So this is some kind of a dilemma, but this is how it works at the moment. And general sample size justification would focus here, as we are in a confirmatory setting, on the type 1 error. And we have here the global type 1 error because we have several tests, so that we want to control the overall type 1 error. And we also have in the sample size calculation a statement regarding the power. Right. So this is a phase, a preclinical phase, where when we really found there an effect, then we would proceed to the next phase. And this would be the phase where the drug is the first time tested in the human body. So therefore, we must be very cautious here, and therefore only a few patients will be included in such trials. And the objective of such a phase 1 trial is only the tolerability and the safety of the drug. And there are some common designs like the 3+3 design, where the sample size is already fixed. And that means that we have here in this scenario a fixed sample size, but we also have to justify the sample size here. And we do this as the sample size is fixed, we have a more exploratory look at, for example, the adverse events and the adverse event occurrence. So we focus on the probability that an adverse event could occur, or we could also look into what information about the safety profile we could obtain when no adverse event is observed during the study of a little few number of subjects. Yeah, the sample size justification here is, I think, a little bit technical, but in the end, it's an estimation of the safety profile we so far get from our trial. If it is agreed on that the drug is can be called safe so far, then we will get into the next phase, in the phase 2. And in this phase 2 study, we are now going into the patients, and that means that we are now interested in the efficacy of our drug. That means we are also interested in dose finding, but we are also more interested, or in our scenario here, we are more interested in the proof of concept. So could we really observe an effectiveness or efficacy in patients? We also use sometimes statistical testing, but the question is more, what information could we obtain from such a study? And therefore we have always the point estimator. This could be, for example, the treatment effect, or when we have just one arm, we could also look at the rate or the rate. And then we have, this is just the point. And then we construct the interval around our point estimator. This is called then the confidence interval. And this confidence interval, or the width of the confidence interval, based on a quantile and of the standard error. But what is important here is that the standard error is depends on the sample size, so that we have a factor that in which we can adjust to make the estimation more precise or not so precise. For example, when we include less patients, then the confidence interval will get wider. On the other hand, when we increase the sample size, we would estimate the treatment effect more precisely. So, and therefore the sample size justification would be more that we say when we do the study and we have this proportions, then we would expect a confidence interval of so many percent, for example. And then we can conclude, or if we can estimate if this precision is high enough so that we get enough information, for example, also to get a treatment effect for a potential phase 3 study. And if we have enough information in a phase 2 study that we have found the correct doses, and we also have a preliminary estimation of the treatment effect, then we will go in the big phase 3 study where the proof of efficacy and safety is evaluated. And in a phase 3 study, it's always a confirmatory proof. That means we are in a confirmatory setting. That also means that we have to use the statistical test, hypothesis testing. And here we really want to show that we can observe a real treatment effect in a real patient population. And we always and what we also do here in this phase is that we want to conclude on the target population, that and therefore it is recommended to use really a randomized controlled trial, because this is the trial or the study with the most evidence from a single, or to get from a single study. Sample size justification here, as we are in a confirmatory setting, highlighted, of course, or focused on the type 1 error, that the type 1 error is controlled, and also on the power. It's very important that there is no doubt that the type 1 error is controlled. This is very important in a phase 3 study. What is also important in all studies, but we have shown this here for the phase 3 study, is that the choice of the primary endpoint. And we are interesting is the theta blocker really able to reduce the average systolic blood pressure in patients who suffer from hypertension. And this could be measured in different ways. For example, we can look at the change from baseline in systolic blood pressure after 30 days of therapy. This would be a continuous outcome. Or we could also define some responders and say, okay, we have a responder, and the responder is defined when we observed a reduction of 10 millimeter mercury in the systolic blood pressure after 30 days of therapy. So we have here a binary endpoint where we just have responders. It could also be measured with a time to event endpoint, that we are looking in the time until the reduction occurred. And what we see here, that we have, based on the chosen or selected endpoint, we have different assumptions we are included. And we have the general parameters here that are almost the same. But what is really important is when we look at the required sample size. We see when we use the continuous endpoint, we need the less patients. And this is because we get the most information about our endpoint when we use a continuous level variable. Because in the binary setting, we will throw a lot of information away. If we, for example, have a subject who has a reduction of 11 millimeter mercury, then this would be a responder, but a subject who have only 9 millimeter mercury reduction would be a non-responder. And here, the information would more be incorporated into the study. Okay, but when the study 3 is successfully conducted and we can reject the null hypothesis and conclude to the alternative, we would get the mark approval, and then we will get in the post-market surveillance. And in this phase, the type of studies are more diverse because we have more things we could focus on. For example, one important point would be the surveillance of the safety, that we look into more rarer adverse events. But what we also want to do is to explore a little bit more the drug and the effect of the drug in some subgroups and so on. So we want to test the drug in a real world setting. And that is the reason why we also have here observational studies or controlled studies. And when we have observational studies, then we have always a problem that we need more complex methods than in randomized controlled trials. And with the higher complexity, we must have, or we have more assumptions that we must make prior to the study. For example, when we have a linear regression here, and just don't want to go into too much detail here, but what we see here is that we need to estimate or make assumptions about 1, 2, 3, 4 parameters, and 2 of them are standard deviations, which is very difficult really to make a good assumption. But there is this rule of thumb that when you need 10 observations for each covariate in a regression model, meaning when you want a linear regression model and you would like to have 2 covariates, then you need at least 20 patients. Okay, when we then have also completed the phase 4 study, then we make a summary of the overall results. And we want to have a summary of the safety evaluation and also of the efficacy evaluation. And this evaluation will not be based on a single study. This will be based on across all studies we have conducted with our drug. For the safety evaluation, as we also included here, healthy volunteers in most cases, we have include all phases, studies from all phases. And for the efficacy evaluation, we will just include the phases where we really test our drug in patients. And this will be done to get a bigger picture of the complete safety profile and of the complete efficacy profile of the drug. So this, in the end, we want to say something about online sample size calculators. And we have an example prepared where we have a responder rate in the intervention group of 98% and a responder group in the control group of 94%. We have a dropout rate of 10% and a maximum type 1 error of 5%, and we want a power of 80%. So with this setting, we go to some online calculators and enter our values and wants to get the sample size. And we have done this in 5 calculators, and we see that we have 5 different results. And these results ranging from 564 to 842. And this difference, I would say it's very significant, because this is a lot of people more we would need when we use calculator 2 than as when we use calculator 1 for the same question we would have. So there is a lot of online calculator available, and honestly, when you have a first rough estimation of in what area we are dealing here, or what area we are looking at here, then this is very helpful to just get an impression, because I know that I will not be able to conduct such a study like here, having 100 subjects. It's more like 500 up to 1,000. But as you can see in the different results, it's not very clear which assumptions are behind the calculator, and also that the methods the calculator are used are often not really provided. So it's always better to have an expert on your team, on your study team, that could provide you with some input for your specific study with a specific objective. And yeah. Right. And the take-home message. So because we are at the end of this webinar, is for the sample size, it is important that the sample size in all studies must be justified. It is not very necessary in all cases that you must have a sample size calculation, but it is very important that the sample size is justified and also matches to your objective. The second thing is that due to ethical reasons, it is important that the sample size is chosen that it is as small as possible, but as large as necessary. And as this often appears when you test not only once, but several times, then a multiple comparison arises directly. And you have to adjust for this when you are in a confirmatory setting. As I said, when you are in an explorating setting, then you can test as much as you want because the type 1 error is not really important here. For the study phase, it is very important that each team member understand the objective of your study and that the sample size you are, the justification of the sample size match to the objective of your study. And as we have seen in the phase 3 study, it is very important to choose the primary endpoint very carefully. And it's always good there also for discussion how the primary endpoint could be chosen, in which way and which scale level we should use here. And in the end, we should you should be aware that some online calculators might be dangerous to use, but for first impression or preliminary look in online calculators are very helpful.
Thank you for watching and don't forget to subscribe to our channel.